So we are going to have Louis Rocha, who is going to tell us about redundancy shapes the dynamical modularity of biochemical regulation for canalization, robustness, control, and evolvability.
All right, thank you very much. So let me start by thanking the members of the group that currently contribute to this, but also those who used to be in the group but already left but contributed to what I'm going to show. So I want to thank them and the sponsors of the work, mostly NIH and NSF for this one, and the FCT in Portugal. All right, so I'm going to start with system science because very nicely Reinhard started by mentioning Ashby on the first day, so I thought entitled to talk about the people I usually talk about. And so the thing is about the world is complex and multi-scale. This has always been a problem from the beginning of the field of complex systems from cybernetics. And perhaps the first person in our field that talked a lot about, in some sense, but not calling it that, modularity was Herbert Simon in his paper on architecture of complexity, which his view of modularity in complex systems here is very associated with perturbation as buffers to perturbation. So he argued from evolutionary principles that due to the need for robustness and noise, modularity is expected in evolution. I would say the argument mirrors an earlier argument in biology, in particular in evolutionary and developmental biology from Joseph Needham. If you're interested, in those days they called this dissociability, not modularity, but the idea was dissociability. So I just thought I mentioned this for historical context. So the key idea of Simon is near decomposability, right? So the key idea that he talks about in his way of thinking of hierarchical modules is this idea of near decomposability, which is the idea that it turns out when you're interested in a given level of observation, a particular level you're interested in, the level above and the level below mostly can be integrated out as boundary conditions to those. So that's what he observed, and most of the variables tend to be near decomposable. Therefore, you might call these holonomic constraints or something like that. That means that you can treat them as effectively in the time scale you're interested as a modular, as a level. So he gave us this good news that even though the world is complex and multi-scale, we can still accessible and study it via hierarchical modular decomposition. So that's the one of the gists of this paper. So I mean, this is not what I want to focus most of it, but we took this idea fairly like about 10 or 15 years ago to study modularity from a different angle in order to address dynamics. So we're very interested in what we call dynamical modularity as opposed to community structure in graphs and so forth. So we converted this idea that Simon talked into what we call perturbation modularity. This is mostly the idea of Artemy Kolchinsky, who was my PhD student at the time. But we wrote this paper about looking at modules or community structure outside of network structure, but as directly looking at dynamical systems. So of course, by this time, as people already mentioned, these plots, network science was alive and kicking, and there was a lot of modularity and so forth that deal with graph representations of multivariate dynamical systems. But what we noticed in this paper is that the graph representations that you do community structure on depend very much on the measure you chose to do the association, what we call dependency measures. And it depends on a number of other things, thresholding, all of that stuff. Also community detection is ultimately optimizing pairwise interactions on these graphs that are difficult to interpret sometimes in terms of the original dynamics, because you had to make all these choices to get there. Also, coupling between variables in dynamical systems does not necessarily lead to significant correlation. For instance, in the paper, we show this example, which is 80 coupled logistic maps, and we show that they have no correlation. They're in chaotic regime and they have no correlation. Yet, using this sort of Simon idea that we develop and as a measure there, in which we look at perturbations of individual variables and how they propagate in time, if you integrate in time, you actually notice very nice modularity that exists in the system, even though they are coupled and chaotically and have no correlations. So we argue this measure in this paper. So basically, this ultimately gives us partitions of near decomposable system variables and integrates in time through time scales, which is rather nice because you don't have community structure at a particular time in time, but you're looking at the measure of overall modularity in time. So that's a nice thing that I think this work. It does not require construction of a network representation. So you sort of querying directly the dynamical system, not the network representation. And ultimately, the idea is Simons. It's separate the intermodular from the fast intermodular from the slow intermodular dynamics, right? And naturally separating that via perturbation spreading, you get naturally the modularity that comes from this. So it captures variation of modular organization across time scales, as I just said, and it's a principal alternative to community detection in networks. So it doesn't have a dependency on a network of pairwise interactions that you might have to do. So this is very nice. I wanted to mention this, though it's not exactly what I want to do. What I want to focus is even this method and all the other ones that we've been talking about in network science ultimately cast modularity as partitions of system variables. And one of the things I want to argue in this talk is that we want to look at modularity not just as partitions of variables, but also partitions of variables in specific states, right? So the modularity is more functional. It's not just the variables, but a variable in a state might be involved in one module, and the same variable in a different state might be involved in a different module. So that's sort of the idea that I want to go. But before I go there, let me give you some more system science ideas from this. So the idea of what we have comes from a follow-up to Simons' paper in this book from 1973 from Howard Patty, who is one of my PhD advisors. He wrote this book called Hierarchy Theory, The Challenge of Complex Systems. And in this, he realized actually systems that are truly complex, they are not near decomposable in Simons' sense. Systems that are truly complex are not reducible to a self-contained multivariate structure. In fact, occasionally, and this is the truth in biology a lot of the time, you have very rare events for which it's even very hard to build inductive data from. Event control, one level controls the other with very rare events. And so even if you integrate it over time, like we do with the previous algorithm that I just showed, you would miss those correlations because they're not correlations, they're control actions. And these control actions can be very rare and unique, but they can change dramatically the dynamics that happens before. So he called these systems control hierarchies, which are different from what we usually think of modularity. So how do we decide this? So more later, one of his students, actually, his first PhD student, Michael Conrad, in his work on evolvability, noticed that in order to study evolvability in biology, you kind of need to match this trade-off between the type of robustness to perturbation modularity that Simon talks to us about, but also this controllability that is necessary to create sufficient variation and enough variation so you cannot be too robust. You have to do this. So he, in this paper, especially the geometry of evolution, breaks up a lot of things, including modularity, though he calls it compartmentalization in that paper, that are needed to occur in biological systems to match this trade-off of evolvability. So one of the things he argues is needed in addition to compartmentalization is different types of redundancy. And so this is sort of what has been the research agenda in my lab for a while, to look at different types of redundancy, particularly in biological systems. And so, I mean, as an aside, you can see how this derives from Patty's work, in that Patty observes in his book that if this is to occur, if we really have control hierarchies, you expect that a higher level that has control over the second requires that there's what he called a selective loss of detail. In other words, you need to forget a lot of the details of the lower level in order to control it. You need to have very simple handles for interventions in the lower level, because you can't control everything, right? When you control a lower level, it's impossible to control everything. So he called this selective loss of detail. And the corollary from this is that if there is emergence of new levels in these hierarchical structures, you're expecting to observe a ton of redundancy at lower levels, so that most actions on the lower level don't have any control action, but many of them do. We could go into discuss this in a long time. I would point that this idea has been picked up in many ways. One recent paper that I really like is the David and John Krakauer and Melanie Mitchell talking about this in terms of emergence in large language models. They argue that these systems don't have true emergence in the sense of PAT because they don't forget stuff. They don't have all the details that they have. So there's this need to forget stuff, which is the selective loss of detail. But that's sort of an aside. We could do this in discussion over beers or something later on, if you want. My point here for modularity is that I want to look at redundancy as something that plays between these two sides as a tuning parameter of having robustness to modularity, in this near decomposability that Simon talks about, but also effective control, even rare interventions that statistical estimations may not pick up. So I look at redundancy as sort of this tuning parameter that does this. And one question is, how do conceptualize modules that capture both of these aspects and the trade-off between them? Okay, so another thing to keep in mind, because I'm not going to go there immediately, is that I really also want to explore this version of modularity, that we're not just looking at partitions of system variables, but partitions of system variables in specific states. So to do that, I have to introduce some of our work in redundancy in Boolean networks. Luckily, you guys are familiar, most of you, with Boolean networks by now, so I'm not going to go into all the details. Let's say I have a Boolean network of one node that is regulated by three others, so its in degree is 3. And then the logic of this system of the variable 4 is given by a lookup table, right? So all the possible combinations of the Boolean values of the inputs give me this. So this, just to use this as one thing that is often forgotten about dynamical systems and Boolean networks in particular, is that an automaton network is not just a graph by any stretch of the imagination. An automaton network is both a graph of pairwise interactions and one of many higher order representations of the causal logic. So in addition to the graph of pairwise interactions, you have a logic that is actually higher order imposed on that graph. And every algorithm that you do that works only on the graph is ultimately not making predictions about this specific logic, but about the full ensemble of dynamical systems that fit that graph. So whether you're using structural controllability or the amazing feedback vertex set theory that you use only on the structure, you are parameterizing the entire ensemble, not just not the specific logic of a particular system. All right, so this lookup table has a bias of 0.25, so has a probability of ones in the output. There is that. It's not very important for the rest of the talk. And so what we do to remove redundancy of these lookup tables, and we've been doing this in a number of papers that are here, is to compute its prime implicants using the Quine-McCluskey algorithm from Boolean logic, circuit minimization. And we substitute or use the hashtag symbol to mean don't care, right? So basically this lookup table is compressed to this one, where the hashtags say it doesn't matter what the value of that variable is. So that exposes the redundancy that exists in those in that Boolean logic. So in truth, what this lookup table is doing is just the logical AND between x1 and x2 and ignoring completely x3. That's what it's doing, right? And so we have measures for this. I'm not going to go into the details, even though this is a mathematical conference. Let's just say that we basically compute the mean number of hashtags in the lookup table under some assumptions. And that gives us a measure of input redundancy, how on average, how many inputs are redundant to that function. So in this case, it's 1.75 on average are completely redundant. And from this, we derive a measure called effective connectivity, which is the in degree minus the input redundancy. So this tells us the effective connectivity here is 1.25. So what this is telling is that even though it's connected to 3 inputs. On average, it decides a state with only 1.25 on average. It doesn't need the other. So there's an amount of redundancy for control. From this, we derive something we call an effective graph, where we resolve the node level effective connectivity to the edge level effectiveness by looking at individual columns on this table. And this gives us a weight. So we convert this original graph, we call an interaction graph, to an effective graph, where this is trying to include a probabilistic parameter that capture how other inputs control it. So one thing to notice here is that these can be collective inputs. As I said, this logic is higher order and the graph is pairwise. So the values that we have here have to, in some sense, are including not just linear inputs into the function. So that's what distinguishes this from the measure of sensitivity and activity that Kaufman and others have generated earlier. Okay. So from an original interaction graph, using this, so this is in the Taliana model, we build an effective graph from this and realize some edges are entirely redundant. You know, they're described here as red, but most of them just have a weighted version of this. So they become, some are much more effective than others in passing signals back and forth. If you want to do this to your models, we have code for this. This is the URL. This is a recent paper with the latest version that Jordan was very involved in doing. You know, I encourage you to look at this. Let me move on. So putting this together a little more, just to give you an example of the type of redundancy you've seen in these models, the agamous gene in this network has an in degree of 9. It's supposedly regulated by 9 other genes, but it turns out it's only on average regulated by 2.1. So these models have typically massive redundancy in them. And you can see their transition tables are full of hashtags, right? So telling you that they are actually quite redundant in what they do. So this is the interaction graph that I already mentioned, and this is the effective graph. We conceptualized this as a probabilistic graph that gives us a causal explanation of how likely perturbation and control signals are to pass, right, through the network. I'm going to go fast through this because it's not exactly what I want to show, but I invite you to look at the page. We look a lot of data of this. We looked at all the models in the cell collective and showed that in biological models, the effective connectivity is much lower than the true connectivity. So there's massive redundancy in experimentally validated models. Many have fully redundant edges. When we compared it to similar to null models, random null models of the same bias and same other parameters, even though the random models also have a lot of redundancy, the biological models have a lot more redundancy than the random models, than the null models, right? In addition to that redundancy, to this effectiveness, they are also much more heterogeneous. Here is the Gini coefficient. I'm going through this quite fast. So it means the cell collective are a lot more heterogeneous in their effectiveness than the random models. So what does that mean? Not only does any molecule or protein gene or whatever in these models have fewer inputs that control it, among them, they're very heterogeneous. Some are much more powerful at controlling than others. Most of them actually have very low effectiveness, but only 1 or 2 are very effective. So they have a very high Gini coefficient. Okay, so we could spend a little more discussing about this, but this is arguably giving you robustness to most perturbations. This is what happens. Not only are they has a lot of redundancy, they are also very unequal in doing that. Going further with this, just to show this is meaningful biologically, when you use the effective graphs rather than the original interactive graph to make predictions about the true dynamics, you realize that most of the time, and I use a counter case and a case here of these genes in this network, the Spearman correlation is much higher when you make your prediction of spreading dynamics via the effective graph than when you do it via the interaction graph. Right? So this means that it's meaningful in predicting the true dynamics. What you have here is the Spearman correlation with the true dynamics when you perturb one node. So spreading dynamics or reachability via the effective graph predicts the true dynamics significantly better than DEG. And you can see how this works, particularly if you start thresholding the graph, you realize that, for instance, some nodes, for instance, there's the Wuschel gene that is predicted to be needed for control, say by FVS and other techniques, it's actually only needed to be controlled to control itself. It's an island of effectiveness in the network. It doesn't propagate any connection. And you can see how the effective graph does a horrible prediction of how this gene correlates with the true dynamics, whereas the effective graph predicts very well that it does nothing on the dynamics, right? So you can realize some of these genes don't do anything on this model, and you come to realize this is not going now. Give me a second. Maybe I'll have to move here. You realize that actually only need the leafy gene to control the entire network. In this network, even though structural control theories predict you need many more genes to be able to control that network, it turns out you only really need leafy. So in another paper in 2016, we showed how structural controllability and minimum dominating set theories are really horrible to predict true control in Boolean networks. Feedback vertex set theory is much better, but as I said in the beginning, FVS is predicting the controllability of the entire ensemble of dynamical systems that fit this interaction graph, not this specific network. When you go to that specific network, you actually have a smaller set of genes that predict. The other one is an upper bound on the genes you need. FVS gives you an upper bound. And so this tells you not only which genes are necessary to control, but it also allows you to see how it actually works. This effective graph actually can tell you how the graph operates, so it helps for explainability as well. I'm going to go very quickly through this. The effective graph has been showing, and some of this is ongoing work. For other controllability, for instance, we use the effective graph for feedback vertex set theory in an upcoming paper instead of using it on the interaction graph. So we look at the strength of the loops in terms of our effectiveness, and we show in various biological models that your prediction of the true controllability versus the predicted by the effective graph is always much better than doing it via the interaction graph, right? It has a lot of assumptions here. But so if you use the effective graph instead of the original graph, you do better. This is not completely straightforward because the feedback vertex set theory requires a non-weighted graph. And so we have a weighted version here to do that, and more details on that will come soon. In terms of modularity, we can also use the effective graph for modularity. I'm going to go fast. In the same paper, we'll present this, but we already, in 21, already had done the weakly connected components. So if you threshold the network according to our effectiveness and compute the size of the connected component, the largest strongly connected component, you realize that most of biological networks, these are all the networks in the cell collective, are quite robust in their weakly connected component, but very fragile in their strongly connected component, which is interesting. So in other words, reachability and signaling and communication is very well preserved with effectiveness, but control of the dynamics and the tractor dynamics is actually more fragile because you need the loops that are in the strongly connected component. It becomes a little more fragile. That's how it looks. So this is a paper that is coming. Now, all of these methods are still looking at modularity as components, so partitions of variables in the original graph. And I want to move beyond partitions of the graph. So, yeah.
So how does this graph look like, say, for random Erdős-Rényi?
From random what?
Erdős-Rényi networks. Like if I just create like a random Erdős-Rényi.
For the effective graph?
Not effective graph. I just want to see like if I create a random weighted Erdős-Rényi graph and then I plot the same graph of like the largest component size versus the edge effectiveness threshold.
Yeah, we haven't looked at that. I could look at that. We looked at null models with different connectivities for criticality using Erdős-Rényi's graph and to look at criticality. So we have those. If I have time, I can show them at the end, but we didn't do that for the analysis of the largest component. But I'm sure it's known because I'm sure people have done that. So we could compare that. Here we were really interested about biological models and see how fragile they are. So, all right. So EG improves analysis over IG or the interaction graph. I still want to do the partitions. To do the different partitions of modularity, I need to introduce yet another form of redundancy. So effective connectivity, you can argue that it's a type of redundancy that is asking things that are not needed or a little needed versus other needed. And now I need to talk about symmetry, which is when you have different things and some can substitute for those. They are equivalent, right? So I already showed you that these lookup tables, in this case, is an example automaton that has 6 inputs, have a lot of hashtags, but I can compress these further by assuming another symbol that is permutation independence, as we call. So this is the little circle we put here, if you can see back in the back, that this whole thing collapses to a single schema that is telling me, as long as I have input 2 and 3 at 0 and 1, and among the other 4 I have a 0 and a 1, this system transitions to 1. So in other words, wherever you have the little circle, it means they can permute with one another, right? So this is a different type of redundancy, which is not possible to put in a graph, in a simple graph. You cannot say in a simple graph that several edges are all together. You need a hypergraph or something like that. We actually use a threshold network to do this type of logic. This is one of the reasons I say the logic of a Boolean network is higher order, because it has relationships that don't fit in a graph, right? So then we come up with a measure of input symmetry. In this example automaton, this has 4 inputs that can change. We have changed the measure of symmetry through the years. The last one with a lot of joined input in this paper. So our last version is from just last year with our code that we have different measures. If you use our code, there's different measures of symmetry, and I can talk to you about those, but I don't want to differentiate them right now. So one thing that I want to see to get to this higher order representation, which we use threshold networks, we cannot put it as a graph. So here, you notice that each one of these schema that we call a two-symbol schema, because it has hashtags and the little squares, converts to a threshold network that tells me the value of the automaton X will be on, represented by black, if you have input 2 off, input 3 on, and among of the other inputs, you have one that is 0 and one that is off. You can see the threshold networks, the merges fiber, and you need to have all these 4 things happening. All these 4 things need to be happening for it to be triggered, and that represents the logic of that particular schema. Of course, then you have the complement to go to off, right? The automaton that has the negation of this tells you what it will go, what the logic that goes to off. So one thing you immediately notice here is that in this network, each variable of the original system is represented with a node in its on state and another node for its off state. So similar to Recalbert's expanded network, right? So you can see that. You can see this. This is a full representation of all the causal dynamics, just a second. And so all the redundancy has been removed. So here, everything that actually moves something is in this plot, and everything that is redundant is out of it. You have a question?
Yeah, quick question. Can you guarantee that for any Boolean function, you always only have one such set of variables that are symmetric?
Yeah, in the 2013 paper, we explained the expression. So you might need 2 layers to explain. For some constraints, you need not just one threshold unit, but one threshold unit that connects to the other, which is the equivalent thing we do here with the merging. So there's a version of this network in the 2013 paper where you don't merge the fibers, and we show that with a 2-layer network you could do anything, including exclusive or whatever parity functions and such things, right?
Yeah, I mean, like, do you need multiple different symbols of circles?
Oh, yes. Oh, that's what you
like
mean.
Is that number of symbols of circles bounded?
I don't want to get into that complexity, but there's different types of group symmetries here. So this one has only a single one. More complicated functions can have separate groups of variables that separately permute. And then you have an index for each one of those, right? That's true. That's sort of described in the paper. It's quite complicated, actually, right? So each system variables are represented for each distinct state. If we go back to this model that I showed earlier, of Thaliana, the agamous gene again, you saw that representation of schemata. So in these plots that we call canalizing maps, every schema here becomes a threshold unit in the DCM. So the threshold units are the blue nodes here that have a threshold value. So let me highlight this specifically. So each one of these threshold units is a row in that table, right? So there's a one-to-one, this is one representation of that logic that says you have to have two items that are on, two conditions to go, and one is a symmetry constraint, the other is not, right? So in other words, every single of the schemata becomes a threshold unit and the associated values in this threshold plot. And this represents fully the non-redundant dynamics. That's an example. This is not as pretty, the plot, because it's the automatic one that our code produces. We want to work on improving how it looks, the threshold networks, but this code that we have here kind of will produce these graphs for you for any logical function that you upload, right? So to put it all together, then we have the interaction graph, the effective graph, and something we call the dynamics canalizing map. This is for a different network, not the Thaliana. This is the canonical Drosophila single cell model from Rick Albert group. This is not the full parasegment, just a smaller single cell model that has 3 input variables or source nodes that control. This is its effective graph, and this what would be its DCM representation. So you can see it looks bigger because every variable is represented twice, one for its on state, one for its off state, plus the threshold units, which represent the schemata. But it turns out it has everything that is redundant has been removed. And so we could explore what we call pathway modules in this, which are dynamical unfoldings, dynamical propagations that are guaranteed to happen if you meet the input requirements. And it doesn't matter what the values of other nodes are. So let me unpack to this. Before I get there, let me say that even though this is more complicated, it actually grows linearly and it's scalable. Because you compute this only for the lookup table of every node. And so the size of this network is 2 to the time of n the nodes you have on the network, plus m, which is the number of schemata in your representation that you have. Okay? So this grows linearly, unlike the state transition graph that which captures all the dynamics, that is 2 to the power of n, but this captures every possible dynamic that can happen as well on this network, right? It doesn't give you attractors, it doesn't give you any of that state transition graphs, but any dynamical propagation that can happen on this network is represented in this DCM. So let me show you a little bit how that works. This is, we go through details. So this network has 3 inputs that I told you about, right? So the sloppy pair gene here, imagine I have this input that tells me the sloppy pair gene is, or protein in this case, it's off, and neighboring wingless protein is off. This is guaranteed to do everything that you see there in blue, that we call a blue module. Doesn't matter what the value of any other variable is in this network. It's completely robust to any value of the other value. So just guaranteeing that you have this happening, you're guaranteed to have this blue module happen, right? So this is what we call a value model. Here, a pathway module. So here is the dynamical unfolding of this network, showing here the state at time 1. It's not showing the input values. So each square here represents a node, an inner node of the network, not the input values. And you can see their grade. They're basically representing the hashtag that I told you earlier about. So they can be in any state. As long as this condition is guaranteed, all of this is going to happen no matter what. This is the blue module. That's why we call this a module, because it's robust to the perturbations of every single other value. Right? So this module, doesn't matter what the other does, will happen no matter what. This is a pathway module of seed set 2. If I add this other input node to off, I apologize that this network actually separates that node in 2. There's 2 versions of this same model. One has 2 nodes, but they're fully equivalent. So this has another module that we call the yellow module here, which makes sure that these 2 are in the state that they are. So in other words, if you have a neighboring hedgehog gene and protein on, the what's in yellow is guaranteed to happen, okay? Which is much smaller than the blue module. It turns out when you have both of them together, they have something we call synergy, that the two of them together light up, as it were, a couple more nodes in this network, right? So these two pathway modules can combine to produce even more, right? And this guarantees conversion to attractor number 3 in this network. It has 5 attractors if you only consider the inside nodes, not the outputs. So this is what we call synergy. These are the 5 attractors. I don't want to go into the details of that. Another example is the pink module that only requires, see, the threshold unit is only 1. That either this one is active or this one is active, not both simultaneously. So this guarantees what we call the pink module, no matter what you do to the others, right? Okay. So this is the point. This is a pathway module of seed set 1. And it doesn't resolve to which attractor you go. It can be one of 3 if you do that. So what I want to show next is how you combine these things. In another circumstance. Now, let's say I combine the pink with the yellow, the pink and yellow module, and then I get all this synergy between them. It grows me a lot more by combining the two. Again, the yellow, but now with the pink, right? And so they go in synergy, and these are all other models. So with modules. So with this, we define something, we call a complex module. Let me go back there, which Akshay showed in his poster in more detail here, and he can tell you about the algorithm to do this, in which we show that the blue, pink, and yellow modules are actually kind of a combinatoric algebra that you can bring every behavior that happens on that network by combining the pink with the yellow or the blue with the yellow. And then there's completely different behavior. So if I do the blue and the yellow, I light up these extra things, the synergy. And if I do the pink with the yellow, I highlight these other ones, right? So these are then what we call a complex module, because it turns out adding any more inputs to these does not expand them. The synergy ends. The synergy ends when I compute the pathway module of seed set 2 with one of seed set 1. But if I go to seed set 4, this one doesn't grow anymore. So that's a context of maximality. And after a while, there's no more synergy in the model, right? So we can look at those details. This is a complex module of seed set 3. Increasing seed set size does not reveal more synergy in this network, because there's no more, in this case, there's nothing else to go beyond this, okay? So there are other modules. There are other pathway modules in the network. I don't want to go into details of that. They're very small and contribute very little to the overall, but some attractors depend on them. You can see basically they are the negation of the yellow module. But what I want to show, and I guess almost finish with this, is that when I project all of these to the original network, the modules are pretty meaningless. Like if I put the blue module here, you notice that the blue and the pink have a lot of the same original variables. But they are, in our way of looking at this, they are different modules because they are in different states. And they actually do very different things. They converge to different attractors. So if I was doing community structure or whatever your favorite algorithm just on the network, I would be collapsing things that functionally are doing very different things, going to different attractors, because I'm assuming that these variables, whether they are in state on or state off, belong to the same module. And what we're saying is variables in a state might be doing one thing, and variables in another state might be doing something entirely different. So if a protein is expressed, it might be participating in a different pathway than if it's not expressed, right? So this is basically what this is saying. If I go to the complex modules to see this, you can see, I'm going to put all the modules there, but the complex modules basically encompass the full network. So The complex module is the full network, and the other one is the full network. But they do entirely different things. One goes to one attractor, and the other goes to the other attractor. So this is what I wanted to highlight, that just looking at the structure and partitions of the original network may be missing a lot of very important functional information in biology, in biological models, because the state matters, the dynamics matters, right? So I will finish now here. I could talk a little more about criticality, but I'll leave it now and can answer more questions. So.
Are there any questions? Couple of questions. You have to switch it on.
Is this pathway module equivalent to this domain of influence from Ricardo's group?
Yeah, it's very similar. I mean, we still haven't done the work to see if it's exactly the same thing, right? So the ultimately it should be pretty isomorphic under certain conditions, depending on how you do the threshold network. So our network is a threshold network, right? So it's been represented in a particular way, with a particular way to do the threshold units, that it's not the same as Recker's network, right? So Recca Albert's network has had different incarnations. The first time that I saw an expanded network was not a threshold network, was a sort of a bipartite graph. Later on, it became a threshold, but by that time, we already had published also our paper with our representation of threshold networks. And so we talk a lot. And so I think there's a paper, I don't know if it came out from Felipe Radicki and Thomas Palmer, a previous student of mine, looking at all of them as isomorphic as threshold networks. But I don't know if that came out or not, so.
A quick question. Yeah. When you were mentioning about near discomposability as opposed to just composability,
I'm sorry, what?
As opposed to composability and near discomposability, what's the difference?
Okay, all right. So one way to look at this is that these modules capture both control actions and robustness. So the pathway module by itself, say the blue module, is very much like a near decomposable approach. It's very robust to perturbation. So you could define it in terms of perturbations. You find the minimum set that is required for it to occur, and it's robust to perturbations on everything. So in a sense, is akin, but not to the same thing, to inferring them via robustness to perturbation. But at the same time, what they do that the others don't is allow us to explain this sort of, lack of a better word, kind of an algebra of combinations that are driven by control actions, right? If I turn on the necessary conditions to do the yellow one, I can propagate that. So one way that I wanted to ask, and that I wanted to do this, but I have a slide here for that. This relates to criticality in the following way. So many people have thought of this trade-off that I talked about for evolvability as a need to be robust and at the same time have a variety of behavior, rich behavior, right? This is often known as the criticality hypothesis. So these things are working on those models that people do this. Say Stuart Kaufman with Boolean networks looks at this as a single layer, as a single level. What it looks from other work that I didn't show here, but Jordan is one of the authors and Recca is also one of the authors, is that the biological networks, at least those in the cell collective, are not at all like that. They are much more stable, right? The networks that are there are very stable, especially if you remove the input nodes or the source nodes from them, if you don't consider the source nodes. So in other words, any type of behavior like critical that you would want to occur has to occur via the control nodes of these source nodes. So what I try to propose as a conjecture here for how these things work and how those two trade-offs come together is that in biological organization, you likely have modules that are super stable, a la Simon, very robust to perturbations, but they have a few input values that control them into completely different attractors. And the combinatories of the control of these inputs at higher levels may give you this richness that sort of the criticality hypothesis is trying to give us, but it's not quite captured. So I think this approach allows us to talk about this because we can talk about perturbation robustness, but also controllability and the few nodes that control this dynamics. So I'm not sure if I explained myself correctly, but I'm happy to talk more about you. Yeah.
Thank you very much for the interesting talk. So I agree. So FES may be too large to control specific systems because it is also a minimum sufficient subset of the variables for controlling systems under any choices of the regulatory functions. But also how we can reduce controlling nodes from FBS so strongly depends on the choice of the functions.
Yeah.
And also I wonder, so how you specify functional form?
Functions?
Yeah, functions. Because also I think so you studies model systems. So you specify, you determine nonlinear functions for each.
I mean, in these specific models that I'm showing here, the functions are pre-specified, right? So these are models that are published and experimentally validated in biological network. I'm not doing anything to the functions. They are already specified. That's why I'm saying the controllability that we infer from here is not the controllability of the entire ensemble of networks that fit a network, but it's of that specific set of logical functions that were experimentally validated. It's one among a huge ensemble.
Okay.
Right.
Yeah. But also, do you agree also their choice of the functions are appropriate? So because also, suppose that one genes may receive k input.
Yeah.
So to specify the genes, so we have to know the input-output relations for two k possible input. And I think so most of the experiment did not do such analysis, and so many mathematical models just assumes functions.
Yeah.
So I think so. Yeah, maybe.
Sure. I mean, and I have to be clear that we haven't compared FES controllability with this dynamics canalizing map. We compared it using the previous representation I showed, the effective graph, because it's a much more easy comparison to do where you have a graph too, right? It would not be entirely fair to compare FES to this. This has the entire causal logic of a particular function, the way a particular model, a particular network. The way I see this is facilitating explanation of why control happens the way it does. And also in some sense, offering a bridge between the micro level of the functions you have at the node and the macro level of the dynamics. This, particularly in this network, it tells us that really kind of a representation that is best for this model is that it has these three, has others, but there are these particular three modules, the pink, the blue, and the yellow, that their combination gives you most of the behavior, most of the dynamical behavior. So that helps as an explanation. So one of the ways that I want to use this is if you have very large models, like say a digital twin model, like the Anna-Niriaki's model, that has more than 1,000 variables of a Boolean network. If we find the modularity, we can collapse a lot of that very large network into these modules and facilitate explanation, right? So that's one of the things I want to do. But any insight of using FES for this is great for me. I mean, so that would be good. So,
thank you very much.
Thank you.
Perhaps we'll just stop there since it's past 5. But let's thank the speaker again.